Oxygen requirements. Oxygen is pretty important, and we know why it's important because most organisms undergo aerobic cellular respiration. Where did oxygen end up in aerobic cellular respiration? Yes, you know that now. It is the final electron acceptor. So being able to take in oxygen is very important for most microorganisms, but not all microorganisms undergo aerobic cellular respiration. And so we can classify different microorganisms based on its oxygen requirements. Does it need oxygen? Does it not need oxygen? So if we classify a microorganism as an obligate aerobe, aerobe, that's oxygen, we get it. They need oxygen. They're aerobic. They use aerobic respiration. They need oxygen for that. If it's an obligate aerobe, if you were obligated to do something or an obligation, then you have to have it. So an obligate aerobe only undergoes aerobic growth, has to have oxygen, and oxygen is required. Now, when I'm talking about oxygen required, I'm talking about atmospheric levels of oxygen, so what's basically in the air, in the atmosphere that we breathe, and this is actually about 20%. And you should kind of know this as comparison purposes as we're talking about oxygen requirements. Okay, obligate aerobes only grow aerobically, and oxygen has to be present in order for growth to occur. We can also classify some microorganisms as obligate anaerobes. So anaerobic growth, oxygen is not used. And because this word obligate is put in front of it, it is has to have an anaerobic environment. In fact, not only can it only grow anaerobically, but if it's in the presence of oxygen, oxygen will damage it, poison it, cause it to stop growing. One example of this is the genome Clostridium. Do you remember when we learned about endospores? Endospores form when conditions are unfavorable to growth and it allows a microorganism to survive by laying down these layers and kind of going into stasis. A lot of organisms that form endospores are obligate anaerobes. They are poisoned by oxygen. So the way they survive is if they find themselves in the atmosphere, they form these endospores. Clostridium tetani is one of those. This is tetanus, causes tetanus. It's an endospore forming bacteria. You find it in the soil. We find it in a lot of places. And it doesn't bother us if we come in contact with it. It's an endospore, but unless it gets inside our body, it's not a problem. It's an endospore, so in the presence of atmospheric oxygen, it's in that endospore form. If we pick it up in our hands from the dirt, we just wash it off, no big deal. Problem is when we get a puncture wound and that Clostridium tetani gets pushed way deep into our tissues where there's less oxygen. Then it will germinate, come out of its endospore, start growing again, and producing toxins that will cause the disease tetanus. All right, so obligate anaerobes, only anaerobic growth, and because it's an obligate anaerobe, if they're in the presence of oxygen, they will be poisoned. And unless they have some sort of way to deal with it, endospore formation, then this organism won't be able to survive. Okay, facultative anaerobes. So anaerobic growth, so we get the anaerobes. Facultative means take it or leave it. They can grow anaerobically, but because it's a facultative anaerobe, it can also grow aerobically. It actually has two types of metabolic pathways, or at least two types. One using oxygen, an aerobic metabolic pathway that allows it to grow, and they also have other pathways that are anaerobic where they can still grow. So this is a take it or leave it. You can actually also call it a facultative aerobe. So facultative anaerobe, it can take or leave an anaerobic environment. Facultative aerobe, it can take or leave an aerobic environment. Both of those are true, but the literature has decided to be consistent, and we refer to all of these organisms as facultative anaerobes. E. coli is a facultative anaerobe. It can grow with oxygen in the lab just fine. It can also grow deep into our intestines where there is much less oxygen, and it grows just fine there too. And that's because it has both pathways, an aerobic pathway and an anaerobic pathway. Oxygen is used, but it is not required, and it doesn't hinder their growth. Now, facultative anaerobes actually grow a little better in the presence of oxygen because they can use their anaerobic pathways either way, but when the oxygen is present, they can also use aerobic pathways, and actually they grow a little better in aerobic conditions. All right, those are facultative anaerobes. What else? Aerotolerant anaerobes. Anaerobes, so we're talking anaerobic growth, these are aerotolerant. If you tolerate something, you don't use it, you don't need it, but you can tolerate. In this case, it's aerotolerant. They can tolerate oxygen. Oxygen isn't poisonous to them, but they don't use it. Doesn't make any difference. These are our aerotolerant anaerobes. We have a lot of microorganisms in this category. They are anaerobic, and they've probably always been anaerobic. They didn't evolve to use oxygen, but they did evolve to have some protections from oxygen. And we're talking about, we'll talk about that in just a minute. All right, so aerotolerant anaerobes, that's another category. There is the last one I want to bring to your attention is called microaerophilic organisms. So microaerophilic, philic means loving, right? Hydrophilic, hydrophobic, philic means loving, aero, so oxygen loving. Micro means small amounts of oxygen. So they only grow aerobically. They do love oxygen, and they do have to have it, but they can only have it in small amounts. And this is not atmospheric. This is 2 to 10% concentrations. If they have too much oxygen, they'll stop growing. If they don't have a little bit of oxygen, they won't grow. So they're in this sweet spot here with just small amounts of oxygen. There's lots of places that have reduced amounts of oxygen that they can do. One example of this is Helicobacter pylori. This is a organism that is actually found in our stomach and can cause stomach ulcers. I think we have a case study that we can look at later for this. Okay, so here's my challenge to you. I want you to look at these 4 tubes. This is a tube of nutrient media, so bacteria can grow in this. This is a loose-fitting cap, so it allows oxygen, which means this arrow represents oxygen in these tubes. Near the cap and near the surface, we have a good amount of oxygen, so high oxygen levels up here. But as we get lower and deeper into these tubes, there's going to be less and less oxygen. You can see the black microorganisms growing here. Look at their growth patterns. I want you to label A, B, C, and D, and then I want you to draw the one that we're missing. So once you figure out which one all of these are, then I want you to draw the one that's missing. Okay, so how'd you do on that one? Let's check ourselves here. This we see growth patterns where we have lots of organisms growing at the top and nothing here at the bottom. What did you label that one? That one is I can't go back very well, an obligate aerobe. Only aerobic growth, oxygen is required, and we need a good bit of oxygen, right? That 20%. So you can see in those tubes that you don't get oxygen growing at these low levels. What about this one? Here, our pattern of growth is where oxygen is not as plentiful. We have very low levels of oxygen, if any, and these are our obligate anaerobes that are growing here. Right at the bottom where there's little to no oxygen is the only place where these can grow. So that's our obligate anaerobes. This one is our facultative anaerobes. Remember, facultative, take it or leave it. They can grow in oxygen. They have metabolic pathways that allow them to grow in oxygen-rich environments. They have metabolic pathways that allow them to grow without oxygen. But you'll notice, where's our density? They are growing better in the oxygen-rich environment. This because they can grow anaerobically anywhere in the tube. Oxygen doesn't hurt them at all. And in fact, they can grow anaerobically and aerobically at the top of the tube. So you get a higher density, a lot more growth occurring there. So this is the growth pattern of a facultative anaerobe. What about this one? Growth pattern is consistent throughout. Oxygen doesn't bother them. That doesn't seem to help them. These are our aerotolerant anaerobes. All right, so what did you do for the last one? I'll draw it on my board on this one. So the last one that wasn't represented was microaerophilic. So if I was to draw a tube that was microaerophilic, and this is my broth levels here. We know microaerophilic can't handle lots of oxygen. We know that it won't grow at the bottom because it needs a little bit of oxygen. Our growth pattern is about here. in that sweet spot where you get 2 to 10%, and you get a band growth right there at that low level. So that would be a representative culture of microaerophilic. All right, so understand these words, understand how that works, and oxygen, and that will help you. All right, let's do nitrogen real quick, and then we'll stop the video. So we get chemical requirements, oxygen, and nitrogen in this video. Oh man, I forgot this part. All right, let's do this part first. So why is oxygen necessary for some and fatal for others? When I'm talking about oxygen, I'm talking about atmospheric oxygen, and we're talking about oxygen, we're usually talking about atmospheric oxygen, covalently bound. This is O2. I remember when we talked to metabolism, we said O2 is not a problem. O2 is two oxygen molecules covalently bound. They're sharing electrons. It's very stable. This oxygen is fine. This oxygen doesn't kill anybody, microorganisms, anything. O2 is very stable. But when oxygen is not O2, when it's singlet oxygen, you remember we talked about they are very good at accepting electrons. They're actually extremely good at, they have a high amount of electronegativity. They are searching for electrons and they will steal them. These are very reactive forms of oxygen. Singlet oxygen is capable of that because they're very good at oxidizing. They're very good at accepting those electrons. And that can be dangerous because they'll take electrons from anything. This is the oxygen that is toxic for some anaerobic organisms, for obligate anaerobic organisms. They do not have any adaptations that protect themselves from singlet oxygen and other forms. Now, there are other forms of oxygen when they combine with things, superoxide, radicals, peroxide, hydroxyl, these are all reactive forms of oxygen. They are damaging. So what do? How do these aerotolerant microorganisms deal with these reactive forms of oxygen? Well, they have to make some enzymes. They have to make something that will help them deal with these reactive forms of oxygen. The one I want you to focus on is actually catalase, because we're going to talk about that in lab. And catalase is an enzyme that we find in our muscle cells. It's an enzyme that a lot of microorganisms have. And what it does is it neutralizes peroxides. Peroxide is H2O2. Peroxide, you may know hydrogen peroxide as an antiseptic. It's in that brown bottle. You get it, and your mom pours it on your cuts, and you see it bubble up. We'll learn about how hydrogen peroxide works, but it is a reactive form of oxygen. It's stealing electrons, which is how it kills microorganisms. That's why it's a good disinfectant or an antiseptic. However, our muscle cells don't want to be killed, and they have an enzyme that they produce called catalase that can quickly take hydrogen peroxide and reduce it to water and oxygen, O2. This is why you get those bubbles when your mom pours hydrogen peroxide on your cut. We don't use this as much anymore because we have found that it's not the best antiseptic. It kills anaerobic bacteria well, but if you put enough of it on there, it can damage your cells too. So it's not used as much. Hydrogen peroxide is not used as much as an antiseptic anymore. All right, so that's an enzyme catalase that does that. There are carotenoids that deal with singlet oxygen, superoxide dismutase. So again, the aerotolerant anaerobes have to have adaptations that let them deal with these reactive forms of oxygen. So that's how oxygen can be helpful and then fatal for other microorganisms. All right. And if you're interested in that, the chemistry involved, you can look at some more details in your textbook. This is that catalase that I was talking about. So you take hydrogen peroxide, H2O2 is the chemical formula for hydrogen peroxide, and this catalase enzyme catalyzes the reaction and it turns it into water and oxygen, and that's where you get the bubbles from that.